Welcome to today's webinar from the International Alliance of ALS-MND Associations, our third annual Global Clinical Trials Update. For this webinar, we have English and Spanish captions available. If you're joining us live on Zoom, please check the chat box as we will be adding the instructions shortly, or scan the QR code on screen if you have another device available. And if you're watching the recording in our YouTube channel, you will find the instructions in the video description on how to activate the subtitles. Para este seminario web tenemos subtítulos en inglés y español disponibles. Si se unen a nosotros en vivo vía Zoom, verifiquen en la casilla del chat ya que estamos agregando las instrucciones en este momento o pueden acceder a la traducción por medio del código QR en la pantalla usando otro dispositivo. Y si están viendo la grabación en nuestro canal de YouTube, encontrarán las instrucciones en la descripción del video sobre cómo activar los subtítulos. For this webinar, we are using automated captions. Please bear in mind that this kind of translation is not perfect and many variables can affect accuracy, and errors are possible. However, even if a few words and sentences are incorrect, you should still be able to get the meaning in context. For those of you who don't know me, I am Jessica Mabe, Programs Coordinator for the International Alliance of ALS-MND Associations, and today we will be getting information on ALS-MND treatments currently on phase 3 clinical trials. Today we are joined by AP Science, Clean Nanomedicine, Ionis Pharmaceuticals, the Instituto de Recherche Farmacológica, and the Stichting Trials Foundation. Thank you all for joining us today to be able to give these updates to our ALS-MND community. Our moderators today are Dr. Nadia Sethi and Dr. Martina DiMaggio. Dr. Nadia Sethi is co-chair of the Northeast ALS Consortium Patient Education and Advocacy Committee. A dentist by training, she transitioned to research-focused ALS advocacy after her husband's diagnosis with the disease in 2019. She serves on the programmatic panel for the Department of Defense's ALS Research Program, the Scientific Advisory Council for the International Alliance of ALS-MND Associations, and the Leadership Board of Genetic ALS and FTD, and the Legacy. She also manages a social media community of over 7,000 ALS clinical trials and research to share scientific content with the patient community. Previously, she served as Director of Community Outreach at ALS TDI. Nadia is currently pursuing a master's in pharmacy with a focus on clinical pharmacogenomics and precision medicine at University of Florida. Joining her is Dr. Martina DiMaggio, moderating this webinar. Martina DiMaggio has extensive experience researching disease mechanisms of amyotrophic lateral sclerosis and frontotemporal dementia and is passionate about making a difference for patients struggling with these diseases. She received her PhD in clinical neuroscience from King's College London and completed her postdoctoral training at University of California. Martina is thrilled to have joined the International Alliance as the first scientific director. Now, some final instructions. Any questions you have, please write them in the Zoom Q&A box. And if you are watching from in the Zoom Q&A box, and you can add your questions also in the comment box in the chat box of Zoom. I also want to take this opportunity to thank everyone who pre-submitted their questions. There is a lot to cover, and we will do our absolute best to answer as many questions as we can in the time we have. I will now turn it over to Nadia and Martina, our moderators for today.
Hello, everybody. Welcome to today's webinar. We'd like to share, first of all, that we tried to extend invitations to everyone who was conducting a phase 3 trial globally, and we're very grateful to all of the presenters that are joining us today in all of the different groups. And then we're going to, as Jess said, do our best to answer as many questions as we possibly can in today's session. And with that, I'm going to introduce our first presentation. We have Dr. Christian Fassot from AB Science, who's going to be talking about the masitinib phase 3 trial.
Hello, my name is Christian Fassot. I'm AB Science Chief Medical Officer, and we thank you for giving us the opportunity to present to you our company and its clinical development program. So AB Science is a late-stage clinical stage French company founded in 2001. The company is based in Paris and has been listed on Euronext, the French stock market, since 2010. AB Science has over 50 employees, mostly dedicated to R&D activities. AB Science has developed two proprietary platforms. The first platform is based on masitinib, a highly selective protein kinase inhibitor in late-stage development for the neurodegenerative diseases, oncology, and inflammatory diseases. The second platform is a portfolio of next-generation synthetic microtubule destabilizers, including AB8939 for hematology indications currently in phase 1. So masitinib is supported by a strong mechanistic mode of action based on neuroprotection. Masitinib exerts a protective effect on the central nervous system by targeting microglia and exerts a protective effect on the peripheral nervous system by targeting mast cells. Masitinib is orally administered. The formulation are tablets in two dosage forms, 100 and 200 milligram, to be taken in the morning and evening. So the development program in ALS consists of AB 10015, a phase 2b/3 study, which has been published, and AB 19001, phase 3 confirmatory study, which is currently ongoing. So masitinib in ALS showed consistent results, including on survival with 4.5 milligram per kilo dose. You have on the right-hand side the results on the primary analysis population of normal progressors with a positive effect on the function, ALSFRS, also on quality of life, on FVC, a median progression-free survival of plus 4 months and a median overall survival of plus 6 months. When we look at a subgroup of patients prior to any loss of function, those benefits are further increased. This is true on the functional scale. This is also true on CAFS, which is a combined assessment of function and survival, which becomes statistically significant. The benefit in quality of life is improved. FVC is further improved. PFS is further improved from 4 to 9 months, and median overall survival improved from 6 to 12 months, becoming statistically significant. And to be noted on the right is that these increased benefits remain positive and valid when you add the fast progressors to the normal progressors. So currently, we have a confirmatory phase 3 study which is ongoing and has been authorized both by FDA in the US and by key European countries. The primary endpoint is the change in LSFRS revised the functional score at week 48, with secondary key endpoints being CAF, progression-free survival, quality of life, and overall survival, as well as biomarkers. This study is facing some slowness in recruitment, and amendments to the design are currently being discussed with both the FDA and EMA towards protocol simplification, namely to suppress the 6 milligram arm, to suppress the run-in of 3 months at the start of the study, to suppress the double-blind extension and to replace it by an open-label extension, to allow enargone concomitant treatment and severe patients, and to focus on a subpopulation prior to any complete loss of function. So when looking at this revised confirmatory study, it will enroll on more or less 400 patients, and the feasibility shows that we could include these patients in 12 months period on 62 sites, so quite feasible study. So last, the masitinib platform offers a late-stage and diversified platform primarily centered around neurodegenerative diseases and also mast cells disease. No compassionate use named patient program are active at this stage. So if you look at the neurodegenerative disease, we are in confirmatory phase 3 for ALS, as I mentioned. We are also in a phase 3 confirmatory study in progressive forms of multiple sclerosis and are ready to start a mild study, phase 3 in mild and moderate Alzheimer's disease. Study has been approved by regulatory authorities. In mast cell diseases, we are in confirmatory phase 3 with mastocytosis and in phase 2 with mast cell activation syndrome. We are also exploring some other indications like sickle cell disease, where we are in preclinical viral diseases like COVID. We have 2 phase 2 studies that are in data reading currently, and in oncology with metastatic refractory prostate cancer with phase 2b3 study completed. So I thank you for your attention, and I remain your contact point and ready, of course, to reply any of your questions. Thank you.
Right. Thank you so much, Dr. Fossat. We now have Dr. Greenberg from Clean Nanomedicine, who will be talking about next phase 3 clinical trial. Joining us for Q&A is Dr. Sfasi.
Hello, my name is Benjamin Greenberg, and I'm a neurologist who serves as the head of medical for Clean Nanomedicine, and I'm happy to be with you today discussing our work in ALS. Tonight's presentation will include multiple forward-looking statements, and the appropriate disclaimer is here, but let's jump into the science. As everyone on this call is painfully aware, there are many factors that can contribute to motor neuron death in ALS. What do we mean by this? So if we look across individuals who suffer from this condition, there are some individuals who are losing neurons because of aggregated proteins within their cells. Some are losing neurons from hyperexcitability, glial activation or inflammation, and or oxidative stress. This is a heterogeneous disorder with both genetic and sporadic forms, and in any given individual, one or more of these pathologies could be leading to the neurodegeneration that our patients suffer from. In the end, what all of these pathways have in common is that they can cause an energy deficit within neurons that leads to cell death. So whether it's that hyperexcitability or inflammation or oxidative stress or toxic accumulation of proteins, science has shown that each of these has the capability of causing an NAD deficit within the cell. Now, NAD is a critical cofactor for the generation of energy within mitochondria. These are the powerhouses of neurons. And when there is a lack of NAD in cells, it has been shown over and over again that they will undergo degeneration. So in a way, the way I like to say this is you could stab, strangle, or shoot a neuron, lots of ways to cause the injury, but at some point it takes its last breath, and that last breath is dictated by a lack of NAD. Now, groups have tried for many years to increase NAD within cells with limited success. What Clean Nanomedicine does is take advantage of a very specific biochemical event known as catalysis. We use a very specific gold nanoparticle whose structure at the atomic level allows for NAD to be created within neurons within a dish, within animal, and within humans. And that increase in NAD is what's associated with survival of neurons. Through multiple different clinical trial programs, we've accumulated more than 700 patient years of exposure to CNM-Au8, and it has been well tolerated, and we have not seen a concerning signal relative to safety. And with this as a background, what we have seen is promising results relative to efficacy. CLEAN has been committed to a variety of clinical development programs, including the Rescue ALS protocol, the open label extension following that trial, the Healy ALS platform, where CNM-Au8 was the regimen C within this platform, and then the open label extension, and multiple expanded access protocols to provide CNM-Au8 to as many individuals as we can. What did we learn from the trials? So there are lots of ways to judge the outcome of a drug, and phase 2 clinical trials are challenging relative to those outcomes. But importantly, we look at the survival of patients. Now, the trials for Rescue and Healy were short in duration for the double-blind period. So one of the things we can do is look to natural history studies and the accumulation of data from those studies to compare to. With this in mind, we use databases for the PROACT controls and the MIND AUS controls to compare the experience of those treated with CNM-Au8 to those individuals who had not been treated in the past. And in each of these comparisons, there was a 50 to 69% risk reduction of death or any cause of mortality. And importantly, what we saw is prolonged survival. When we look at individuals within these cohorts, we work to match them to like patients. So we take individuals who were in the rescue trial and compare them to the same age, sex, duration of disease, and look to see if they survive longer on CNM-Au8 compared to those controls. The same analysis was done with the Healy ALS platform, and here we saw a 48 to 57% reduction in risk of mortality when compared to these historic controls. Summarizing the survival data we have from different groups, Rescue ALS, Healy ALS, and our expanded access protocols, in each of these we have seen a statistically significant difference between individuals treated with CNM-Au8 and those who were in placebo controls for prior trials, supporting the fact that CNM-Au8 led to improved survival in individuals who were treated. Importantly, beyond survival, in Rescue ALS, we were able to see that there was a delayed time to clinical worsening. This was defined as time to mortality, tracheostomy, assisted ventilation, or the need of a feeding tube to be placed. And this composite outcome, when comparing individuals who were treated with placebo versus those who were treated with CNM-Au8, in the Rescue ALS platform, we saw a 71% risk reduction of clinical worsening for those treated with CNM-Au8 compared to those with placebo at 36 weeks. Within the Healy ALS platform, over the 24-week double-blind period, we saw a 74% risk reduction in time to ALS clinical worsening. So in each of these trials, Rescue ALS and Healy ALS, we were able to show not only survival, but delay in worsening. Armed with this data, CLEAN is committed to a global phase 3 trial in ALS, which we refer to as Restore ALS. This would be a randomized placebo-controlled trial in a 2-to-1 randomization, where for every one individual in placebo, 2 would be randomized to CNM-Au8, and this would indeed be a global trial. The objective would be to investigate the effect of CNM-Au8 on survival and delayed clinical worsening events in ALS. We would enroll individuals who were within 36 months of onset and had adequate respiratory function. They would randomize in a 2-to-1 fashion and be followed until an open label extension launched. The primary endpoint of this trial would be survival, but importantly, there are multiple key secondary endpoints that we would be examining. The ability to delay the need or prevent the need for permanent assisted ventilation, as well as tracheostomy, non-invasive ventilation, feeding tube initiation, and any additional support that may be needed. Likewise, we would be following multiple functional scales to determine whether or not CNM-Au8 would prevent decline in addition to expanding survival. Importantly, with this trial, it would allow for background treatments to be continued, and if new therapies were approved during the course of this trial, individuals would be allowed to take those therapies and remain in the trial. Like any trial, it's important to get data relative to the pharmacokinetic and pharmacodynamic characteristics of the drug. So, blood work would be incorporated into this trial, not just to understand how the drug was metabolized and how it affected the nervous system, but to look at key biomarkers of neuronal survival. And of course, we would have a focus on safety as well. So, our next steps are to engage with regulatory agencies in the United States and throughout the world, plan and hopefully initiate the phase 3 trial, and collect and report our long-term ALS experience through our ongoing expanded access protocols. Thank you for your attention. If you have any questions, feel free to email us at info@clean.com, and my colleagues are happy to take your questions. Thank you.
Thank you so much, Dr. Greenberg. And next up, we have Dr. Stefan Miller from Ionis discussing the Fusion trial. And we will have Rob Goldstein Mahone joining us for the Q&A at the end of the session.
Hi, I'm Stefan Miller from Clinical Development at Ionis Pharmaceuticals. I'd like to thank the Alliance for giving me the opportunity to talk about the Fusion study. This is a study Ionis is conducting for people living with FUS-ALS. FUS-ALS is ALS caused by mutations in the fused in sarcoma gene. This is my disclosure slide. I am an employee and shareholder of Ionis Pharmaceuticals. FUS-ALS is ALS caused by mutations in the FUS gene. It's very rare, approximately 1% of all ALS, and it occurs both in inherited familial ALS and in sporadic ALS. So about 3 or 4% of familial ALS and probably less than 1% of sporadic ALS. The most unique thing about FUS-ALS is that it's the most common ALS-causing mutation in juvenile and pediatric ALS. So these are the most common known mutations in ALS with a young onset. The FUS-ALS presentation varies widely, both in symptoms at diagnosis and also in age of onset. So the onset generally earlier than the overall ALS population, but the age of onset varies incredibly from patients who have been reported as young as 11 years old and patients as old as 80 at onset. Another unique thing about FUS-ALS is when it occurs in the youngest patients, it's often associated with a very fast progression. So FUS, what is FUS? FUS stands for fused in sarcoma, and it's an RNA binding protein that's very important in RNA function and turning RNA into protein. Ionis is testing a drug called ulefnersen, and this cartoon shows how ulefnersen works, what its mechanism of action is. So neurons have the genetic code in DNA, and the DNA gets turned into RNA, and then protein is made from that RNA. So people with FUS-ALS have a mistake in their DNA in the FUS gene. They have a mutation in the FUS gene DNA, and that error is carried over into the RNA, and then that causes not the correct normal FUS protein to be made, but instead a FUS protein that doesn't do its normal jobs with RNA, and it instead aggregates or clumps in the cell body, and somehow those clumps are toxic to motor neurons and cause motor neurons to die. So in the blue there, this is the drug ulefnersen that we're testing. It's a type of drug called an antisense oligonucleotide. And ulefnersen works by binding to messenger RNA, just the FUS messenger RNA, and causing it to be broken down so that that abnormal FUS protein is not produced. And then ulefnersen reduces that toxic function. So the hypothesis is that degrading these mutant transcripts will reduce aggregation of the FUS protein and reduce motor neuron degeneration and slow or maybe even stop disease progression. This is the FUSION study design. FUSION is a phase 3 study with a 72-week placebo-controlled double-blind treatment period. Patients are randomized 2-to-1 to ulefnersen or placebo. So 2/3 of the patients get on ulefnersen and 1/3 are given a matching placebo. After completion of that 72-week period, patients who wish to continue can all receive ulefnersen. So open label treatment with no blinding is available for up to 5 years. The study drug, ulefnersen or placebo, is administered via lumbar puncture or spinal tap every 12 weeks with an additional loading dose after the first 4 weeks. Assessments, many assessments in this study, either in the clinic or through phone contacts at home, the ALS Functional Rating Scale, ALS-FRS, measurements of breathing capacity, measurements of muscle strength, and many other questionnaires to assess physical function and quality of life. There are many safety assessments also. This is the first study in ulefnersen, so it's most important to determine whether it's safe. So there are MRIs, ECGs, a number of different safety labs on blood samples, urine samples, and cerebrospinal fluid, and of course, a recording of all adverse events. It's a small study as far as phase 3 ALS studies go, with a planned randomized population of up to 85 patients. Here are some of the key eligibility criteria for entry into the trial. Patients have to have an approved mutation in the FUS gene. They have to be older than 10 years of age with no maximum age limit. They have to have signs or symptoms of ALS, and they cannot already have a tracheostomy or already require ventilation of more than 22 hours a day. And a full list of eligibility, there are many others, as can be found at that clinicaltrials.gov link there. Ulefnersen is being evaluated on top of standard of care, so any other background therapy like riluzole is allowed. Tofersen or Qalsody is not allowed. This map shows the 24 sites that we have in 16 countries, primarily US, Canada, and Europe, but also 3 countries in Asia and also Brazil. Enrollment began in mid-2021 and is expected to complete next year, likely early next year. And there are just a couple other things I wanted to say about. Ionis provides travel support to patients in this study, so if a patient is identified who does not live near one of the sites, Ionis will pay for travel, even flights, with the caregiver, hotels, so that the patients can be able to participate. And Ionis also has a sponsored ALS genetic testing and counseling program. So this is not just for FUS, but it's a 33 gene panel done through Prevention Genetics, and your doctor can order this test, especially for people who do not have insurance coverage for it. It's very helpful. The eligibility criteria are shown there, and along with the testing, there is genetic counseling before and after. Here are a number of links for additional information. And I want to, of course, thank the patients who are participating in the trial and their families, and our investigators and site staff. And thank you to the Alliance for giving me this opportunity.
Thank you, Dr. Miller. Next up, we have Dr. Elisabetta Pupillo from Istituto di Ricerche Farmacologiche Mario Negri, and she will also be joining us for Q&A at the end.
Morning to everybody. I'm Elisabetta Pupillo, and I work at Mario Negri Institute for Pharmacological Research in Milan, Italy. Today, I would like to present to you the main characteristics of a new clinical trial that will start in the next months, and the title is a randomized phase 2-3 trial on the biological and clinical effects of acetyl L-carnitine in amyotrophic lateral sclerosis. This study received a financial support from the Fight MND Association, and it will be sponsored by Mario Negri Institute and the University of Sydney, Australia. Carnitine is the primary transporter of fatty acids across the mitochondrial membranes. ALCAR, that it is the acronym of acetyl L-carnitine, is the acetyl ester of carnitine, and it is a donor of acetyl groups and increases the intracellular levels of carnitine. In ELS mice, there were performed several studies where we were able to observe that ALCAR counteracts the motor neuron death induced by toxic agents or deprivation of trophic factors. Also, ALCAR is able to slow the disease progression in animal models, ameliorates mitochondrial dysfunction, restores the synaptic transmission, and exert protective effect against neuroinflammation. So we can observe that it decreases the oxidative stress markers and pro-inflammatory cytokines. A previous phase 2 clinical trial was performed. We enrolled 80 subjects, and we detected a significant difference between groups active and placebo at the end of the study in all the primary and secondary endpoint. We performed also a retrospective case control study where 180 subjects were included, but in this observational study where we compared the administration of acetyl L-carnitine with two different dosage, 1.5 grams or 3 grams, we were not able to detect any differences in none of the selected endpoint. Based on this observation, also promising, the study results are insufficient to support a claim of efficacy of ALCAR. The clinical effects of the drug were not accompanied by the investigation of biological markers of the pathophysiology of the disease in the previous trial and also in the observational study, and no study verified an adequate dosage to be used in ELS. Based on this observation, we decided to apply for a new clinical trial, because there's an urgent need to document the effects of treatment on some markers of the disease mechanism and efficacy of two different dosages. So we decided to organize a phase 2 free multi-center randomized double-blind placebo-controlled parallel group trial in participants concurrently taking riluzole. We will enroll 246 subjects. The subjects will be divided in 3 groups. Each group will be composed by 82 subjects, and subjects will be randomized to receive the 1.5 grams of ALCAR, 3 grams of ALCAR or placebo, and the treatment will be administered each day for 24 consecutive weeks. This is the visit schedule. So the treatment period is 48 weeks. We will perform a visit number 1, where the subject will be screened and randomized. followed by a second visit 4 weeks after the screening visit, then a third visit at 12 weeks, then fourth visit at 24 weeks, fifth visit at 36 weeks, and then the final visit, the visit number 6, that will be performed at weeks 48. The subject will remain on treatment during the entire duration of the treatment period, of course, at the same dosage. Case of dropout, an end of study visit must be scheduled as soon as possible to document the study discontinuation reasons and the subject evaluation study. Which is the expected timetable of this trial? Australian competent authority approved the study in April of this year. The Italian competent authority will approve the study more or less within the end of this year or in January 2025. The enrolling will start in next month, in this month, in November 2024 in Australia, while in Italy it will start in January or February, according with the approval of the study by the Italian competent authority. As you would like to receive the additional information about this study and or the course of this study, you can go to clinicaltrials.gov website and search the study number NCT 06126315, or you can send an email to me, to my email address, or to Steve Vucic, who is the study coordinator located at Sydney. I would like to thank you for your attention, and here available for your questions.
Thank you so much, Dr. Pupillo. And next up, we have Dr. Michael Van Ness, who is a neurologist and the head of neuromuscular department at UMC Utrecht. And he will be talking about the trial of lithium carbonate called the MAGNET trial.
I will be presenting the MAGNET study, which is a phase 3 study on the efficacy of lithium in a genetic subgroup of ALS patients. So the genetic subgroup we're targeting for this trial are patients who are homozygous for a genetic variant in the UNC13A gene. And the UNC13A gene was first identified through genome-wide association studies as a risk factor for ALS. Now, subsequent studies have shown that this polymorphism in the UNC13A gene is also a potent modifier of the ALS phenotype. So we see that patients who have this genotype have more frequently have cognitive involvement, more frequently have bulbar onset, vital capacity is lower at diagnosis, and unfortunately, disease progression and survival are shorter in this population. Recently, it has been shown that this effect on the phenotype is caused by a loss of function of the UNC13A protein. So there's a cryptic exon in the gene, and in the presence of TDP43 pathology, this exon is included in the gene, causing nonsense-mediated decay and therefore no functional expression of this gene. Now, we became interested in the UNC13A gene and lithium as a potential treatment for it as a result of a study we performed a number of years ago. So the objective of this study was actually to look at whether genetic variants that influence survival in ALS could potentially also affect the outcome of clinical trials. And to analyze this, we used data from previously performed studies with lithium in ALS. And we used the lithium studies because there were quite a number of them, which resulted in a large data set, which permitted us to perform these post hoc analyses. So what we saw is indeed that the UNC13A and C9orf72 groups had poorer survival than the overall population. And much to our surprise, we also saw that the patients with UNC13A who were treated with lithium appeared to have a better survival than those who received placebo. And there was approximately a 30% difference in the 12-month survival probability between these groups. And this is an effect we did not observe for the patients with C9orf72 repeat expansions. So this effect prompted us to perform a phase 3 confirmatory study, and that study is now underway. So in this study, survival is the main outcome measure, and all the analyses are event-driven. So that means 41 events will trigger a interim analysis, and the final analysis will be performed after 72 events have occurred. At present, we have randomized 60 patients, and we think we will need 171 patients in total to be randomized in this study. We estimate the stop of recruitment will be in the July of 2025, and that we will have the final results in October of 2026. The study is performed at 15 sites, or we're aiming for 15 sites. One still remains to be initiated, but that will happen soon. The study is running in a number of countries in Europe and Australia. So it's in the Netherlands, Belgium, Sweden, Spain, the UK, and Australia. And you can see the sites on the slide there. In terms of other programs associated with this trial, there is an open label extension, which patients can opt to continue in after they've gone through the follow-up period. We would like to perform additional analyses on CSF, but we recognize that undergoing lumbar punctures is burdensome, so participation as part of the study is optional. And then we also have a electrophysiological sub-study using CMAP scans, which we're performing and hope to analyze as an experimental outcome measure in this study as well. So I'll leave you with my contact details and those of Anne-Marie Janssen, who is the project manager for this study. And I would also, of course, like to thank the funders of this study, which are the Cherry Landsfrauen Foundation, MNDA, ALES Stichting, VitaMND, Ullekara, Lindqvist, FWO, and Luzon.
All right, we can proceed to our Q&A session. If the speaker would come on camera. All right, welcome all. We've had, as we mentioned, we've had a large number of questions coming through, so we will do our best to address as many of them as we can. Unless, Nadia, you want to start, maybe I can start with the first question and then we can go ahead from that. One of the questions that came through so from the US is, do you have any suggestions for gaining EAP access to drugs that people do not qualify for in trials? That's for anybody who wants to take this question.
I can start maybe.
Thank you.
We have ongoing EAP programs, so hopefully that will be helpful. Clean Nanomedicine has several intermediate expanded access, or as you would more commonly know them, compassionate use programs. I believe we have the largest in the ALS community thus far. Two of them were initiated by Clean, First one with Mass General, second one through our own expanded access program, about 18 sites. And most recently, we were awarded through the Act for ALS funding, a $45 million grant for an additional EAP program that should be able to enroll approximately 180 patients. So yes, there is opportunity to enroll in EAP programs across the U.S. For CNMA U8. And we do have a website that's dedicated to the most recent NIH awarded to share where the sites are. There is also a remote option for those that meet those criteria. We have now PALs that are residing in Alaska and in very other remote parts of the country. So yes, there is opportunity.
Thank you so much. Anybody else would like to comment? Otherwise, we can just go to the next question.
Dr. Sapasi, would you be able to maybe elaborate on that a little bit and share how that EAP data will work into your development for your drug?
Yeah, I mean, I think we hope as we gain the data from the expanded access programs that we will be able to utilize and better understand how CNMA U8 works in a much more global scale. As you can imagine, for our phase 3 programs, we have a very, very small sliver of the population that is taking CNMA U8 or any other product in a phase 3 clinical trial. But when you have an expanded access program where it's sort of the rest of the population, if you will, you gain a lot of real world experience regarding your product. And so what we hope to do is have that sort of go hand in hand with our phase 3 program data as we prepare our overall package and overall understanding of CNMA U8, both in phase 3 programs, but also within the clinical trial spectrum, and then ultimately discussing with regulatory bodies. I think what's important that's shifted in the expanded access program arena are these new, the new NIH funded Act for ALS, funds that come for EAP, because we're conducting the expanded access programs much more like clinical trials than we have ever in the past. So, in the past expanded access programs, because the aim has always been to get product into as many people, folks living with disease as possible. We don't technically monitor for efficacy as such. It's usually safety, so that we can spend the appropriate rest of funds on obtaining product. But the new programs are much more focused and driven like clinical trial programs. So that data should be really helpful and help us move the needle into how we look at real world evidence.
Thank you so much. That was very helpful. I don't know if anyone else had anything to add. I'll give you guys a minute and then or a few seconds, and then we'll jump into the next question.
Yeah, I guess the only thing I'd add, Nadia, for our listeners around the world, this topic goes by many names, and I would just offer that it's really important that if this is a topic that's interesting to you, that you speak to your doctor about it. In most cases, a physician is the individual who initiates a request for expanded or early or compassionate access, whatever the phraseology in your region is. I just wanted to provide some education that if you're interested in this topic, it's best to begin the conversation with your healthcare provider.
Yeah, thank you, Rob. That's an excellent point. I guess on that topic of expanding access to populations and areas of the world that may not have access to these trials, Can you or anybody else who would like to expand on how is your organization actively, reaching out to those areas that are not, normally involved in clinical trials, and how you are expanding your trials to be global for real?
Well, I'm happy to start and offer our perspective at Ionis. We are a global company, and we aim to operate that way as best we can. We have team members located, in many different locations around the world that are contributing to the advancement of our research, and we have collaborators as well located all around the world, working to advance research on FUS, ALS, and other topics. I think some of the work of the Alliance is worth lauding here on trying to draw attention to the broad community interest in underserved populations in areas like Africa and elsewhere. And it's important that that work continues so that regions like those continue to develop the needed resources and capabilities to execute clinical trials. I would just say that it is important to note that each clinical trial comes with its own design and challenges. Not all investigational medicines are developed and delivered in the same way. Some trials may require different capabilities than others, and that may be a limiting factor in some cases as to how broad access exists. In closing, I would just say that often clinical trials offer travel support, and cross-border participation is something that our community has a lot of experience with. On that topic, to provide education, that often is a decision up to the local institution, and is made possible under local laws on how individuals from outside one country may be able to access investigational medicine trials in another country.
Yeah, thank you for that great comment. I guess we had a question coming in from Colombia on that topic of what would be the practical steps that you'd recommend somebody living, for example, in Latin America, to be able to access any of the trials that you've just talked about. If there is any guidance from you, from anybody in this panel.
Yeah, I mean, it starts by talking to your doctor again. I think it's like, I cannot stress enough, it is your physician who is your best advocate for you. They understand your individual medical history, and they can help navigate often complex situations, especially when that comes to traveling outside of a country. So my advice there is to start to talk to your doctor about your interest in clinical trials. Go in with a printout of the trial, help give them time to get educated on it, so that they can provide, an opinion on whether or not they share your interest, that it's appropriate. And they may be able to also provide you some resources on how to get insurance, let's say, to travel overseas, or to kind of start to help you understand what some of those travel restrictions or barriers may be in the country in which you aim to participate.
If I could just add to that. So we here in Utrecht do have some experience with patients coming in from other European countries to our facility to participate in clinical trials. I think that's a good thing. We also do see that traveling for clinical trials is something which is very demanding. So it's quite a large undertaking. So I think one of the things to look at as well is what does participation actually mean? How feasible is it if you need to travel long distance? So clinical trials which would require someone to come in very frequently, I think are not suited. So it would have to be, I think, something where you would come in and say every 3 months or so, where you could, 3 or 4 visits, maybe that's it. Would be more suited than say early phase studies where sometimes people need to provide blood samples every few days or weeks. The travel demand, I think, has to be realistic because it is very challenging.
Thank you so much for that. I think I'm going to shift a little bit. We had lots of questions. We talked a little bit about expanded access or ways to access treatments kind of outside of a traditional trial. But we've also gotten some questions about trial inclusion criteria and why things are kind of set at the 2-year mark in many cases. So would any of you be able to share a little bit about what goes into choosing inclusion criteria for your trial? Why would you choose a specific patient population?
Yeah, so I'd like to comment on that. So in general, people designing trials, I think, have tried to include patients with similar rates of progression. So your placebo cases are comparable to those who are getting the active compound. The other thing is that from just purely a statistical point of view, patients who do not progress during the period of a study, it's very hard to show any effect, right? If nothing would have changed, even if you wouldn't have given an experimental drug, then it's hard to show your drug is working. The other thing, unfortunately, if patients are progressing very rapidly and a compound would take some time before effect becomes evident, then somebody who would pass away within a few months maybe is not, the compound doesn't get sufficient exposure to show that it's working. So from sort of a perspective of study design, that's very important. Obviously, that's very harsh to patients. If you want to be in a study, that means you might get excluded. We've tried to get around that in a way, using a different approach. So the classical approach is to say, people over a certain age or a certain disease duration or a certain vital capacity. And what we've done, if we've designed a risk score, so we call it the NCALS or TRICALS risk score, which allows you to combine a number of disease characteristics, which put you into a total score, and patients with comparable total scores appear to have similar disease progression. And using this risk score is actually more inclusive. So I don't know off the top of my head, but I think it allows up to 40 or maybe 50% of patients, more patients, to be eligible for clinical trials than using fixed cutoffs, or things like vital capacity above 50 or 60% or an age above a certain limit. And we've actually also gotten advice on that from the EMA, and they've actually endorsed this. So I think we're moving away a little bit from having these fixed cutoffs, because we know that the things that influence disease progression, it's not just one thing, it's multifactorial. So somebody who has gotten to a vital capacity of 49% over the course of 2 years might not be rapidly progressive, while somebody who gets to 49% in 4 months is rapidly progressive. So even though they have the same vital capacity, they're not the same type of patient or not having the same type of disease progression. So I think a multifactorial risk score is probably more useful, but I think that also needs to be developed further.
I think, Michael, you pretty much gave examples that really when you talk product specific for us, at least for CNMAU8, you're absolutely right. And in designing the phase 3 program, knowing that our product, for example, takes minimum of 17 to 20 weeks to reach a steady state where we start to see efficacy changes, that plays a huge part in making sure that those that are enrolled are able to reach that time point in order to have what we expect those efficacy results to be. Also, when you're looking, as Ben presented on our slides, at survival, if folks are not progressing, and it sounds very morbid to say this, but if it goes to your point of making sure that we have a population which statistically meets that sort of enrollment criteria, but also at where we hope the trial will end. If you're looking at survival and no one in your program is progressing, you're never going to finish a program. And these are quite large trials at this point when they're global. So I think to your point specifically, really ring true for us as a company as well. So I completely agree.
I concur, of course, with the comments that have been made regarding the science, which remains the basic for designing your trial. So, I mean, your eligibility criteria have to be consistent with the drug mechanism of action, first of all. But on top of that, and of course, all the clinical criteria that have been mentioned. But on top, there are regulatory constraints that come on top of what you would like to do or what you thought to do about your trial. And sometimes regulators have different views than patients, than clinicians, and then pharmaceutical companies. So you have sometimes to to compromise a little bit for regulatory purposes. And an example in the phase 2b/3 we had, and in the first phase 3 is regarding the fast progressors, and we were asked by the authorities to mimic the real world of ALS, if I may say so, by not excluding the fast progressor, which of course was not ideal in the design. So one way to go around that was to consider this request as an exploratory one, and we added 10% of fast progressors to the whole study population. So there are ways around, but I mean, there are also constraints that are not always, I would say, unfortunately, scientific and clinical one.
Oh, if I can add something, sometimes we have also to add an exclusion criteria according to the safety profile of the drug, because we have also to evaluate if there's something that can worsen the health state of the subject, including in the study, and we must be sure that the drug will not provide additional effect that can improve the health the status of the subject immediately, both because it can have an effect on the endpoint of the study, but first of all, in order to save the safety profile and the safety for the enrolled subject.
Yeah, I mean, in a clinical trial, in a clinical development program, the main goal remains to get the product registered and so on the market, meaning available for the most possible patients, possible in the shortest time possible. So there is a trade-off also between that goal, that is to reach, the availability as soon as possible, and the allowance of inclusions, perhaps of some subpopulations that might, if not delaying, at least complicate the whole development. So there is no ideal situation there, and of course especially when you are patients, you would like to participate to any single possible trial, but so unfortunately not possible.
And if I could just add, again, to help educate the audience here, oftentimes trials start with one set of inclusion/exclusion criteria, and there are occasionally changes to that as trials move forward. For example, in the fusion trial, we altered the minimum age to participate down to as young as 10 years old, as you heard from Stefan earlier. So I just want the community to know that there is opportunity to update inclusion exclusion criteria, but as the rest of the panelists said, those updates still have to meet the minimum from a medical and scientific rational perspective, and they must be aligned with regulatory bodies as well. It is really important in closing to say that the community have a voice in this. I think a couple of the panelists mentioned that this is not just a group of scientists sitting in a room in isolation. When we develop protocols, they are done in a collaborative manner with community members, physicians, doctors, regulators, and others. They're not meant to just be something that's put out that's not community informed.
Yeah, thank you so much. I love the engagement on this question. Thank you for participating. To answer this last point, I guess talking about subject population, we had a few questions on, stratification and what is used to, at this point, to stratify people that participate in a trial, and what kind of are the readouts that are most often used for that kind of analysis. So I know this is a big topic. So anybody who wants to go first, I'm sure there's going to be a lot of discussion on it.
I think it's very study specific, first of all. So there is no one common answer. And of course, for international trials, you stratify usually on geography, starting by that, and then on co-medications. So there are different levels, and your stratification strategy, if I may say so, is very dependent on what your study is about. And what you have learned from previous trials as well.
Yeah, so I think so there's only a limited number of things you can stratify for because there's statistical penalties to pay for it. So I think it depends on the study and what you think is most important for it. I think one of the things that's probably relevant is also dependent on what other drugs are approved in your region. So for instance, in the European Union, edaravone hasn't been approved, but it is approved in the US, for instance. So that seems an important co-medication which you want to take in account. So it also depends, because in the magnet study we're not stratifying for it, but it's run in Europe and Australia, so it's not a co-medication which we're encountering there, so we're not using it. But if you're running a study in the US, I think that would be a very important thing to take into account. So yeah, I don't think there's one universal answer to this.
Would you be able to elaborate on what it means that there's statistical penalties? Just explaining that a little further for our audience.
Yeah, so what you're starting to do is you're starting to divide your population into subgroups, right? So you're saying, I want to be sure that my primary outcome wasn't influenced by, so you wouldn't want to have a study, say, with all males on placebo and all females on active drug, or all your, all the placebos from Australia and all your active patients in Argentina or something. So you want to make sure that certain things are balanced, right? But so you can't really balance everything. So there's certain things you leave up to randomization. So by chance, and if your study is just large enough, that'll balance out. But there's certain things you don't want to leave to chance. So you don't want all the patients to be in Argentina and all the controls to be in Australia, for instance. So that's not, so you're going to say there needs to be balance there. But because you're doing this, that it also involves additional analyses, and the more analyses you do, the more chances you're introducing of having false positive results as well. So that's the statistical penalty you're paying. So you can stratify for 2 or 3 things, but beyond that, it becomes your study becomes very hard to perform. But then also the statistical penalty becomes larger, also making the chances of a significant result smaller. So you need to pick what's relevant and just do those things.
Thank you so much for that. Anybody else would like to comment on stratification specifically?
Well, yeah, just despite the penalty that has just been explained, despite that, nevertheless, at least pharmaceutical industry tend to have too many stratification factors. So a common feedback we get on protocols from regulators is to reduce the number of stratification factors for the reasons mentioned. But despite the fact that this is costing something in terms of study robustness and number of patients, etc., because we are interested on those factors, we would like to know, but we have to keep it to a reasonable number.
Yeah, it makes sense. Thank you so much.
All right. I would love to. I think that this panel is absolutely very interesting because we have so many different types of drugs here. And I think it's very reflective of our global pipeline right now in ALS. We did get some questions about genetics. Dr. Van Ness, if you can maybe talk a little bit about UNC13A and how it's the risk factor and how common is it in people with ALS?
Yeah. So it's a I think a little bit of a complicated story, but so it's a risk factor. So it means that if you carry this genetic variant, your chance of developing ALS is larger, but it's not directly disease causing. So it's not the same as the FUS mutation, right? So that if you have that, then your chances of developing ALS are very large, maybe for some of them up to nearly 100%. So this UNC13A variant increases your chances of developing ALS by about 1.3. So your lifetime risk of ALS is 1 in 350. If you have this, it goes up to 1.3 in 350. So it's a genetic risk factor, but it's not causing the disease to be in your family, right? So relatives of people don't need to be worried excessively that they also will develop ALS. Now, the thing is, if you do have this, and then in your motor neurons there is TDP43 pathology, then the splicing of this gene gets changed and leads to a loss of expression of these genes in those cells, right? And that causes an additional problem in the cell, and that's why the disease becomes worse if you also have this. And I think that's one of the things, a number of pharmaceutical companies, but also the objective of the magnet study is to try and correct this effect. And we see that roughly 1 in 6 ALS patients carries these variants in UNC13A. So we're hoping that if this proves successful, either lithium or a different approach, that we would be able to provide something useful to about 1 in 6 patients. And we've looked at the geography of this genetic variant in different populations. It seems to be relatively stable throughout the world, in at least in the populations for which there's large data sets available. So there's no real differences between populations in North America, Europe, Australia, and Asia, as far as we can see. but we don't have access to large data sets from South America or Africa.
And Rob, I think on that note, do you have any, are you able to share any information about the Ionis genetic testing program, and how would people even know if they have an FUS mutation?
Yeah, I can make a couple of general comments. First, thanks for the question. I think the ALS community and field, when we look at rare disease, is kind of one of those communities that's leading the way in genetic testing overall. I mean, the community has built just tremendous awareness, of the role of genetic testing and helping people make, informed decisions about clinical trials and other topics. And it's globally available. I mean, there's genetic testing and counseling for many ALS genes available in many different pathways, around the world. And the community should be really excited because there are new genetic discoveries being made all the time. As you know, Nadia, if you put up that graph of, we discovered the SOD1 gene, I think in like 93 or 91. But then everybody realized there was more, and we continue to discover more genes that are both thought to be pathogenic or related or otherwise all the time. I'm being a little bit redundant here. I mean, the first step is to talk to your doctor about it. I mean, the decision to get genetic testing or go through genetic counseling is not a minor decision, and it comes with both unintentional outcomes and intentional outcomes. So it's really important that people kind of understand what genetic testing is, what the results may mean, how they may impact themselves and other people in their life. The sponsored testing program available from Ionis is an option. And if, somebody wants to talk to their doctor and they want to pursue it, you can find information about it on our website, and give your doctor information, and they can order the test online that way. I would just say, finally, like, the time to get results can vary a little bit, and to set expectations for people pursuing genetic testing, it can vary. Sometimes you can get a genetic counselor in a couple of weeks. Sometimes it takes a couple of months to get that genetic counseling session done. But we would say it's really important to make sure that genetic counseling is done together with actually the test itself. I hope that's helpful. And I'll say, from the clinical trial perspective, and Dr. Van Ness, please add on, if you're going into a clinical trial and the clinical trial inclusion or exclusion criteria has something about a specific gene being known to exist, usually that trial process will include verification of that, which can include up to doing additional clinical testing if it's needed. And those costs are usually borne, through the trial as part of the screening process.
Yeah, that's exactly right.
Yeah, fantastic. Thank you. That's definitely a great opportunity. So I'm just wondering, for the rest of the treatments that are less targeted and might be more for the broader population, do you have any comments on, whether you have any hypothesis of whether it might affect some part of the population more than other, or general comments regarding accessibility and how people can be involved in these trials? If they have you want specific characteristic, of course there is. That's what we just talked about, but I just wanted to hear from the other people on the panel on this topic.
As far as masitinib program was and is concerned, we did not exclude SOD1 patients from the first phase 2b3 trial. So we had some patients potentially with mutation there. We didn't, of course, analyze that specifically, and for the confirmatory phase 3, we are not excluding either for the time being.
Thank you. Dr. Pupillo or Sebastian?
Encourage anybody who has a large study to look at the specific genetic subgroups if they have that data. I mean, the SOT1 and FUS, I think in the general population are quite rare, so less than 1-2%. So even in a study with 300-400 participants, you're likely to have more than 10 individuals carrying those mutations. So I think as a subgroup analysis, that probably doesn't make sense, but the C9orf72 is more common. So that would be interesting. And I think looking at variants that influence survival, such as UNC13A, are also interesting to analyze as a group. And then,
or even, oh, sorry, keep going.
yeah, so and then the last thing is I mean, also consider how your drug is metabolized. I mean, if there's potentially ultra rapid or slow metabolizers of whatever your compound is, then that might also cause people to either have side effects or drop out or not show efficacy if they just really rapidly metabolize whatever you're giving them.
Or even in terms of like looking at genetic cause versus non-genetic cause generally without going into the specifics of single variants, might be interesting. I just wanted to give Dr. Pepillo or Dr. Sabasi the last chance to comment if they'd like.
Oh, yes, thank you, Martina. Now, in our study, we will not request specific criteria, also according to the genetic test, but we will collect data in order to evaluate at the end of the study if it's possible to divide subject for genetic mutation or not, because we need a good amount of subject with a specific mutation in order to perform the statistical analysis. And in case, we will provide some hypotheses of an effect of the drug or not, also related to a specific mutation. But as inclusion or exclusion criteria, there's no necessity about this. Thank you.
Thank you.
A general comment is that it is obvious that data mining is really underused, but this is a resource issue to start with, and I think at individual company level or institution level or whatever, there are limited resources, both human and financial, that could be apply to explore many things that are just out there probably. So the kind of consortium approach, not only between industry but across all stakeholders, and of course starting with alliances like yours, is I think very important for the future, and even from failures. There have been, unfortunately, plenty the last weeks and months. Even from failures, there are data, and data are exploitable. So, I mean,
Absolutely.
A lot of things to do there, I think. But yeah, it's a complicated field.
Yeah, I love that comment so much. Thank you so much, Dr. Fassa. Dr. Sabasi, do you want to add anything?
Yeah, I mean, for us specifically, the genetic component, because as someone mentioned earlier, the mechanism of action, as we consider CNMAU8, we don't anticipate that there will be a genetic component to therapy, either in favor of or against CNMAU8. So for our phase 3 program, there will not be any sort of component that is analyzing genetically. And, as much as we would like to learn it, because as Christian said, financially, it's a big undertaking for us, especially since we know the mechanism of action for CNMAU8 is unlikely to have that sort of being able to categorize based on genetic mutations. So it's not something that we would undertake at this time in our studies. And that's just by nature of trying to spend the money where most appropriate to be able to conduct the trial. I mean, in terms of finances for a trial like ours that is, north of 500 or 550 patients, we're talking $75-80 million in itself to conduct a study. And just funding that overall in itself is quite a challenge to begin with for smaller companies.
All right. Well, I think this is a great topic to close our conversation on. We'll want to thank you so much again for joining us and for having these very insightful conversations. Unless Nadia, if you want to comment further, or I'll pass it on to Jess.
No, I just thank you so much for being here today. I think this information is so crucial to get out to the ALS community. And yeah, I'm just grateful for this opportunity and for all of you guys to come and share about your amazing programs for ALS.
Thank you. Thanks, everyone.
So yes, thank you very much to all of our panelists. We're so grateful for your dedication towards finding a treatment towards ALS MND. Thank you for spending this time with us today. And I also very special thank you to our moderators, Dr. Sethi and Dr. DiMaggio. Thank you for helping, manage all the questions. And that brings me to a special thank you to our audience for joining, for participating, for sending your questions. That's what made this webinar possible, everyone's contributions. So I want to wish everyone, wherever you are in the world, a great morning, afternoon, or evening. And thank you all very, very much.
Goodbye. Thank you.
Thank you to everybody.
Bye. Thank you.
Bye.